In order to better explain the nervous control of the GI tract, I would like to make a quick review of nervous system function. Remember that the nervous system collects information. The structures within the nervous system that collect information are called the receptors. Receptors in the GI tract will be cells that can detect chemical changes, which we call chemoreceptors, pressure changes, which will detect whether the organ is full or expanded. We can call those mechanoreceptors. Receptors in the GI tract are going to monitor the state of the GI tract as far as their chemical composition and the state of the organ as being stretched or relaxed. This information is going to be sent to the central nervous system. The central nervous system consists of the brain and its spinal cord. The response will be carried out by effectors. Effectors were organs innervated by the nervous system that responded to nervous stimulation. There are two categories of these organs: muscles and glands. In the case of the GI tract, the muscles are going to be smooth muscles of the organ of the GI tract, smooth muscles of the stomach and the intestines, for example. In the case of glands, in the GI tract, we'll find digestive system glands, digestive enzymes glands, I should say, mucus secreting glands, buffer secreting glands. These two types of effectors are going to be innervated by the autonomic branch of the nervous system, also called the ANS. You should remember from last semester that the parasympathetic stimulation of the GI tract stimulated the GI tract, whereas the sympathetic stimulation of the GI tract inhibited the GI tract. Also remember that the parasympathetic neurotransmitters was acetylcholine, whose acronym was ACH. The sympathetic neurotransmitters were epinephrine and norepinephrine, whose acronyms, actually, the neurotransmitter itself was norepinephrine, whose acronym is NE, but sympathetic stimulation also caused the secretion of sympathetic hormones, epinephrine and norepinephrine. One last point. The GI tract has its own plexus of nerves, which combined are called the enteric nervous system, or ANS. Mechanoreceptors and chemoreceptors also send information to this enteric plexus, which in turn is going to respond by stimulating the secretion of the glands and contraction of muscles. So the GI tract is receiving a stimulation from the central nervous system and its own enteric nervous system. A stimulation coming from the central nervous system is called the long reflex, whereas a stimulation coming from the ENS itself is called short reflexes. The word reflexes denote that this is autonomic. These are autonomic responses. So the GI tract is receiving a stimulation from two sources. The central nervous system via autonomic nervous system motor neurons, and the enteric nervous system, which is local. The effects of a stimulation will be carried out by two sets of effectors, digestive glands and a smooth muscle. A smooth muscle is located in two places, muscularis mucosa, which causes local movements, and muscularis externa, which causes more profound movements, peristalsis, and segmentation. The response can be either a stimulation or inhibition of digestion of digestive glands, or a stimulation and inhibition of movement of a smooth muscle. And I should say and, or, because both could happen at the same time, or just one of those two effects can happen. This PowerPoint summarizes the concepts that we have been talking about. Remember, the first step is a stimuli. The stimuli is going to be detected by chemoreceptors, mechanoreceptors within the GI tract. But remember that there's also external stimuli that can affect the GI tract. Things like sight of food, smell of food, taste of food, the thought of food. These kind of stimuli stimulate the central nervous system. The central nervous system, in turn, via the ANS, is going to stimulate the nerve plexuses in the GI tract and the parasympathetic division of the ANS is the branch that stimulates the GI tract. The effect is going to be contraction of the smooth muscles and secretory activity. By the same token, the receptors found in the GI tract itself, chemoreceptors, osmoreceptors, and mechanoreceptors, will also stimulate nerve plexuses. These nerve plexuses refer to the submucosal plexus and the myenteric plexus. In turn, the effect will be a smooth muscle contraction and secretion of glands. Remember that the response of the GI tract is going to be that of secretion and movement. Regulation of GI tract activity involves either a stimulation or inhibition of these two activities, secretions and movement. Acetylcholine, which is the neurotransmitter secreted by the parasympathetic nervous system, stimulates secretion and movement of the GI tract. On the other hand, norepinephrine, which is the neurotransmitter secreted by the sympathetic nervous system, is going to inhibit secretion and movement of the GI tract. The neurotransmitter used by the enteric nervous system neurons is serotonin. Serotonin stimulates the GI tract into secretions and movement. Some of the hormones and parahormones secreted within the GI tract that influence its activity are substances like gastrin, which is a hormone secreted by the stomach. Gastrin stimulates the activity of the GI tract, specifically the stomach. So the stomach will begin to secrete and contract. Secretin, on the other hand, secreted by cells in the small intestine, are going to inhibit GI tract activity. Paracrine chemicals are chemicals secreted within tissue, not within blood. These chemicals have local effects in the cells around them. One of the most important paracrine chemicals that influence the GI tract is histamine. Histamine is secreted by some of the cells of the organs of the GI tract, like the stomach, for example. Histamine stimulates GI tract secretions. The serous membrane that lines the surface of the abdominopelvic cavity and covers most organs within it is the peritoneum. As all serous membranes, the peritoneum is a double-layer membrane. The visceral peritoneum extends over the surface of most digestive organs. The parietal peritoneum lines the walls of the abdominopelvic cavity. The peritoneum of the abdominopelvic cavity is a large complex membrane which folds, creating double-layer membranes called mesenteries. The function of mesenteries is to provide a route for blood vessels, lymphatics, and nerves to travel into the organs. Mesenteries also hold organs in place and store fat. A few organs within the abdominopelvic cavity are not covered by peritoneal membranes. These organs are called retroperitoneal organs. Retroperitoneal organs of the gastrointestinal tract are the pancreas and the duodenum. You can see in this illustration the location of the retroperitoneal organs, pancreas and duodenum. These are retroperitoneal organs of the GI tract. The rectum in the pelvic cavity and the urinary bladder in the pelvic cavity are also retroperitoneal. The membrane in blue is the parietal peritoneum. The membrane in orange is the visceral peritoneum. The purple color structures, which are the folds of peritoneum, are the mesenteries. You can see that the liver, the stomach, the small and large intestines are part of the organs that are covered by peritoneum. Incidentally, the duodenum is part of the small intestine, as we will see later. So, not all of the small intestine is covered by peritoneum. Most of it is. The mesenteries are divided and named according to their location. The lesser omentum and the greater omentum are the mesenteries attached to the stomach. The lesser omentum attaches the stomach to the liver. The greater omentum folds as an apron in front of the intestines. The mesentery proper are the mesenteries are the peritoneal membranes that attach the small intestines to the walls of the abdominopelvic cavity. On the other hand, the mesocolon are the sheets of mesentery that attach the large intestines to the walls of the abdominopelvic cavity. In this illustration, you can see the lesser omentum that falls from the stomach to the liver. The greater omentum will be a sheet of mesentery that falls in front of the intestines as an apron. In this illustration, the greater omentum has been cut. This illustration shows the lesser omentum, and it also shows another mesentery called the falciform ligament found in the liver. This illustration is a nice depiction of some of the mesenteries. The greater omentum has been lifted so that we can move out of the way the large intestine, and we can see the mesocolon, which is the mesentery that attaches the large intestine to the walls of the abdominopelvic cavity. Mesentery proper are the mesenteries that attach the small intestines to the walls of the abdominopelvic cavity. The serosa of the stomach is the visceral peritoneum. Remember that the external muscularis externa of the stomach has 3 layers as opposed to 2 in other organs of the GI tract. The 3 layers will be the oblique layer, the middle circular layer, and the inner longitudinal layer. Remember that the stomach has no submucosal plexus. Also, the mucosa of the stomach is folded into folds called rugae. Rugae increase the surface area of the stomach and form non-permanent folds that will iron out as the stomach fills with food. The lining of the stomach, simple squamous epithelial tissue, is going to fold into stomach glands or pits. These stomach glands or pits have a population of cells that are involved in secretory activity of the stomach. The population of cells found in the lining of the stomach and in the stomach glands are mucous neck cells, which secrete mucus. Mucus is an alkaline product that buffers the acidity of the stomach. Parietal cells are going to secrete intrinsic factors and hydrochloric acid. Intrinsic factor is needed for the absorption of vitamin B12. Chief cells secrete pepsinogen, which is an enzyme or the precursor of an enzyme called pepsin, involved in digestion of proteins. Finally, there is a population of enteroendocrine cells at the bottom of the stomach pits or glands. These enteroendocrine cells are going to secrete hormones into the blood and paracrine factors into tissue and lumen. Both hormones and paracrine factors are going to influence the activities of the stomach. Enteroendocrine cells of the stomach are going to secrete paracrine factors and hormones like histamine, which is one of the paracrine factors that stimulate parietal cells. Parietal cells in turn will secrete hydrochloric acid and intrinsic factor. Gastrin is a hormone. Gastrin is going to stimulate parietal and chief cells. Parietal cells will again secrete intrinsic factor and hydrochloric acid. Chief cells secrete pepsinogen. which is caught by hydrochloric acid into the working enzyme pepsin. Pepsin is involved in the digestion of proteins. Finally, somatostatin is secreted by some of the enteroendocrine cells of the GI tract. Somatostatin is going to inhibit the function of gastrin and insulin secretion. So somatostatin is going to stop gastrin secretion. In stopping gastrin secretion, somatostatin will stop secretions by the parietal and chief cells. Parietal cells, especially those in the pyloric region of the stomach, will secrete intrinsic factor. All parietal cells in the stomach secrete hydrochloric acid. Food that arrives at the stomach is called bolus. The bolus of food mixes with the stomach secretion and turns into chyme. Hydrochloric acid acts on the chyme by deactivating salivary amylases. Amylases are enzymes that digest carbohydrates. Hydrochloric acid destroys these enzymes, which means there is no digestion of carbohydrates in the stomach. However, hydrochloric acid activates pepsin. Pepsin is the enzyme secreted by chief cells. Pepsin digests proteins. Therefore, digestion of proteins takes place in the stomach. Hydrochloric acid further digests proteins by denaturing them. Another function of hydrochloric acid is to kill bacteria. Parietal cells secrete hydrochloric acid. Chief cells below secrete pepsinogen. As pepsinogen travels through hydrochloric acid, it will be caught by hydrochloric acid into its working form called pepsin. Pepsin digests proteins, including itself. So once pepsin appears, it can synthesize its own digestion, creating more pepsin. Gastric secretions go through three phases: the cephalic phase, the gastric phase, and the intestinal phase. The shortest of these phases is the cephalic phase. The cephalic phase starts when we think, smell, or taste food, which is going to stimulate the central nervous system into stimulating the parasympathetic vagus nerve, which will in turn stimulate the stomach. Histamine secreting cells and gastrin secreting cells will be stimulated. Histamine causes the parietal cells to secrete hydrochloric acid. Gastrin is a hormone that will travel through the bloodstream, causing parietal and chief cells into secreting their products. The next phase is the gastric phase. The gastric phase starts when food arrives at the stomach. When food arrives at the stomach, mechanoreceptors will be stimulated as the arriving food stretches the stomach. That stimulation is going to cause stimulation of the ENS neurons, which in turn will stimulate histamine and gastrin-producing cells and will stimulate the myenteric plexus to begin peristalsis and segmentation. Histamine will cause an increase in the secretion of hydrochloric acid. Gastrin stimulates parietal and chief cells, Parietal cells will further secrete hydrochloric acid and intrinsic factor. Chief cells will secrete pepsinogen, which will be turned into pepsin, and digestion of proteins will start. Notice that if the pH decreases below 2 due to the actions of histamines and gastrin stimulating parietal cells, this will inhibit parietal cells and will cease hydrochloric acid secretion until the pH increases to a less damaging value. This mechanism prevents overacidity of the stomach. The next phase in gastric secretion is the intestinal phase, and this is the longest phase, lasting between 2 and 4 hours depending on the contents of the meal. The phase begins when food arrives at the first part of the small intestine called the duodenum. The duodenum is the location of chemical and physical digestion. The duodenum is small, it's about 6 inches long, so it will not hold the entirety of the contents of the stomach. So small portions of the contents of the stomach will arrive at the duodenum, extending, stretching the duodenum, and lowering the pH of the duodenum. These two conditions are going to stimulate enteroendocrine cells in the duodenum into secreting their hormones, secretin and cholecystokinin. The acronym for cholecystokinin is CCK. Secretin and cholecystokinin are going to inhibit the stomach into secretions and movement. So secretin and cholecystokinin inhibits stomach secretions and stomach movement. The stomach ceases their actions, giving time for the recently arrived food in the stomach to digest. A carbohydrate-rich meal will not stimulate CCK, which means gastric phase will continue, making the chyme in the stomach pass quickly from stomach into duodenum. On the other hand, a meal rich in fats and proteins stimulate cholecystokinin secretion, meaning the stomach will stop, giving more time in the duodenum for these foods to be digested. The organ that follows the stomach is the small intestine. The small intestine consists of 3 portions: the duodenum, the jejunum, and the ileum. The duodenum is a small portion of the small intestine. It is the first portion adjacent to the stomach. It consists of a retroperitoneal portion about 6 inches long. Following the duodenum is the jejunum. Following the jejunum is the ileum. The ileum is the last portion of the small intestine, which connects with the first portion of the large intestine called the cecum. The duodenum is the location where most chemical and physical digestion will take place. The duodenum is intimately associated with the pancreas and the liver. Notice that the duodenum receives bile from the gallbladder via a common bile duct. It also receives digestive enzymes from the pancreas. Adaptations of the small intestine increase its surface area. The permanent folds of the small intestine are called plica circularis. The permanent folds, in turn, are made by smaller folds of the lumen of the small intestine, of the lining, simple columnar epithelial lining, called villi. Both plica circularis and villi are going to increase the surface area for food absorption. Further increasing the surface area of the small intestine, of the surface of the small intestine, are microvilli. Villi are the folds of the lining of the small intestine made by simple columnar epithelial cells. Each individual cell in turn folds its cell membrane, creating what's called microvilli, further increasing the surface area for absorption and secretions. The total area of the small intestine, when the plica circularis, villi, and microvilli are accounted for, equal the surface area of a football field. Each villi contains capillaries that are going to absorb the digested food substances, and lacteals, which are lymphatic system capillaries that will absorb digested fats. So to recap, the mucosa of the small intestine is adapted to increase surface area. The plica circularis is made by folds of the lining of the small intestine called the villi, which further is folded by each individual simple columnar cell into microvilli. The surface of the small intestine consists also of infolds of the lining called crypts, where the enteroendocrine cells will be found. These are the cells that secrete hormones like cholecystokinin and secretin, among others. Capillaries, blood vessel capillaries and lymphatic capillaries will be found in the villi, absorbing nutrients. Goblet cells, found among the simple columnar cells, will secrete mucus. In the submucosal portion of the small intestine histology, there will be duodenal glands, which will secrete buffers that will counteract the acidic pH of the chyme arriving from the stomach. And finally, the small intestine will be protected by lymphoid tissue that will contain numerous immune system cells. One of the main functions of the liver in regards to digestion is the production of bile. Bile is involved in the digestion of fats. It is also an alkaline product, which is going to buffer the acidity of the food arriving from the stomach. Bile is produced by hepatocytes, liver cells, sent via the right and left hepatic ducts into the cystic duct for storage and concentration in the bile. So the gallbladder stores and concentrates bile. Duodenal hormones like cholecystokinin and secretin are going to stimulate the gallbladder into releasing bile during digestion bile will exit the gallbladder, travel to the cystic duct, common bile duct, and through the hepatopancreatic ampulla be secreted into the small intestine. The right and left hepatic ducts, right and left hepatic ducts combine to make the common hepatic duct, which are going to take bile through the cystic duct into the gallbladder. The gallbladder stores and concentrates bile. When bile is needed for digestion of fats, bile will exit the gallbladder via the cystic duct, travel through the common bile duct, and be secreted into the duodenum. Pancreatic secretions of digestive enzymes will also take place, and they will secrete their digestive enzymes via the hepatopancreatic ampulla into the small intestine. Secretion and production of bile takes place during the intestinal phase of gastric secretion by the actions of hormones secreted by the enteroendocrine cells of the small intestine. These hormones, secretin and cholecystokinin, are going to influence bile production and release. Secretin is going to stimulate the liver into producing more bile. Cholecystokinin is going to cause contraction of the gallbladder and the release of bile into the small intestine. About 90% of the bile secreted into the small intestine is going to be absorbed at the end of the small intestine in the ileum, will circulate in blood, and when it goes back to the liver, it will stimulate production of even more bile. The location of the pancreas is intimately associated with that of the duodenum of the small intestine. The head of the pancreas abuts the duodenum. The body of the pancreas follows, and the last part of the pancreas is called the tail, which is in the direction of the spleen. The pancreas has two functions. The endocrine function of the pancreas involves the formation of hormones, glucagon and insulin. These hormones are involved in blood sugar regulation. The exocrine function of the pancreas involves the production of digestive enzymes. Digestive enzymes are produced by acinar cells of the pancreas. The digestive enzymes are collected by the main pancreatic duct, which joins the common bile duct, and the digestive enzymes will be delivered together with bile by the hepatopancreatic ampulla onto the duodenum. Digestive enzymes of the pancreas includes pancreatic amylases, which will break down carbohydrates, pancreatic trypsin and pepsins, which will break down proteins, and pancreatic lipases, which will break down lipids. A stimulation of pancreatic secretions takes place via the vagus nerve, parasympathetic stimulation, which causes secretion of digestive enzymes. Secretin, on the other hand, will stimulate, which is one of the hormones secreted by the small intestine, is going to stimulate the pancreas into secreting a buffer of bicarbonate ions. Cholecystokinin will also stimulate the pancreas into secreting digestive enzymes. The large portion of the GI tract is the colon or large intestine. The first part of the colon is connected to the last part of the small intestine, which is the ileum. So the ileum empties its contents onto the cecum, which is the first part of the large intestine. The cecum, in turn, will bring the contents of the colon through the ascending colon, the hepatic flexure, transverse colon, splenic flexure, descending colon, sigmoid colon, and rectum, and finally the anal canal. Defecation will take place at that point. Peristaltic movements brought in by parasympathetic stimulation via the vagus nerve of the rectum causes defecation. An overview of digestion shows that carbohydrate digestion begins in the mouth via the action of salivary amylases. Carbohydrate digestion will stop in the stomach because the hydrochloric acids of the stomach will break down the salivary amylases. Digestion of carbohydrates will pick up again in the duodenum via the actions of pancreatic amylases. The broken down carbohydrates in the forms of disaccharides and monosaccharides will be absorbed by the epithelium of the small intestine. Lipids will not be broken down. There will not be any significant breakdown of lipids until the small intestine, where the actions of bile salts made in the liver and secreted by the gallbladder are going to break down fat molecules enough to be acted upon by pancreatic amylases. Broken down fats in the form of fatty acids and monoglycerides will be absorbed by lacteals. Protein digestion begins in the stomach by the action of pepsin secreted by chief cells. The broken down polypeptides will be further broken down into smaller molecules by the actions of pancreatic trypsin, chymotrypsin, and peptidases. These peptides will further be broken down into individual amino acids by the action of brush border enzymes secreted by brush border cells in the epithelium of the small intestine. So these peptidases will further break down peptides into their individual amino acids, which will be absorbed by the small intestine. Reabsorption of water takes place in the small intestine. Liquids have been accumulated through the GI tract by ingestion, by the production of saliva, production of gastric secretions, pancreatic secretions, bile, and small intestine secretions. Most of these fluids will be reabsorbed back into the blood by the small intestine. The large intestine will also be involved in this reabsorption of fluids. About 1% of the fluids produced during the GI tract during digestion will be excreted in the form of feces.